Okay, this time we'll talk about haemoglobin. So just a reminder, this is all to do with transport in animals. So how, for example, are we transporting oxygen around the body? What adaptations do we have? And one of those adaptations is the red blood cells themselves. They've got their biconcave shape. They've got no nucleus, so it can maximize the amount of oxygen that they can carry. And then within the within the red blood cell, they are carrying millions, over 200 million haemoglobin molecules. So, little recap from what you've done with biological molecules. Haemoglobin is a protein, and it has a quaternary structure. So, if you remember, there's the primary, secondary, tertiary, and quaternary. And that's something you can look back on later on. We'll just focus on the quaternary part. So it has a quaternary structure because it has more than one polypeptide that makes up this whole molecule. So there are 4 different polypeptides. There's 2 alpha helix. There's one there. There's another. And there's 2 beta pleated. One there and one there. And the other part that makes it quaternary is it has prosthetic groups, which are non-protein parts added to it. And the prosthetic groups here are the heme groups. And there's 4 heme groups, one attached to each polypeptide chain. And these heme groups are made up of the iron. Okay, that should be in uppercase too, Fe2+. Okay, right, so this blood cell carries millions, or over 250 million haemoglobin molecules, and each haemoglobin molecule can carry 4 oxygens. So you can see how one blood cell could carry, around 10 million oxygens just by itself. So really efficient way of carrying oxygen in just one blood cell. So when we talk about haemoglobin, it's quite a few things to talk about. We're going to talk about something called the haemoglobin dissociation curve. And then when we talk about haemoglobin dissociation curve, we have to talk about partial pressure of oxygen as well. And we'll talk about this conformational change as well. All right. So first thing to state is these heme groups are found close to the center of the haemoglobin molecule. So if we were to do like some sort of 2D image of the hemoglobin, the heme groups are quite close to the center. So for oxygen to get to these heme groups, it's going to have to diffuse quite a big distance to get right into the center part. Okay, so that'll all be explained a bit later why that's important. Right, so what is the role of haemoglobin? Well, its job is to carry as much oxygen as possible. And it does the job really well. It does it better than just your, if blood had no haemoglobin at all, the blood wouldn't be able to carry as much oxygen. So what it needs to be able to do is pick up, we use the correct terms here now, so you might think of, you could say haemoglobin picks up oxygen at the lungs. But I don't want you saying that. Instead, I want you to say it associates. So haemoglobin associates with oxygen. The opposite being, it could, if it's going to pick up, we're not going to say it anymore, we're going to say haemoglobin associates with oxygen at the lungs. And then the other one we can say is haemoglobin dissociates with oxygen at respiring tissues. So these are two very different environments. The lungs, by their design and ventilation, always have a lot of oxygen in them. And we say that when there's a lot of oxygen, it has a high partial pressure of oxygen. So when you see high PO2, that means high partial pressure of oxygen. There's a lot of oxygen. So in terms of things like diffusion, there's a high concentration of oxygen. Whereas, so that, the heart is going to pump blood to the lungs with a high partial pressure, and this is where the blood is going to pick up, or the hemoglobin is going to associate with the oxygen. And then when the blood moves away from the lungs and ends up at respiring tissues, and I've used the biceps as an example because they're muscles, they respire a lot more than most other tissues because they work hard. But it applies to all tissues, but just these ones are more oxygen hungry, if you like. So these have always got a low partial pressure of oxygen because as soon as oxygen arrives to these regions, it's needed for respiration. Oxygen plus glucose produces CO2 and water. So this region here is always low in oxygen, and this region here is always high in oxygen. So we'll talk a bit now about how the hemoglobin is good at picking up oxygen or associating with oxygen, and how it's really good at dissociating with oxygen when it needs to let go. Right, so this is the hemoglobin dissociation curve. And if you were to put any other liquids like water into different environments of different oxygen pressure, partial pressure, you would expect to see a direct proportion here. As the partial pressure of oxygen goes up, you'd expect the saturation of oxygen in water to go up as well in a straight line, a linear relationship. Whereas what's happening with hemoglobin is it kind of starts off like a normal liquid, but then it shoots up really quickly. Hemoglobin is becoming saturated with oxygen at much lower partial pressures than a normal fluid would. So if you look, for example, if we look at 4, at a partial pressure of 4 kilopascal, saturation of a normal liquid might be what? 25%. Whereas at the same partial pressure, hemoglobin is nearly at 60% saturation. As we go up even more, it's, by the time we get to a partial pressure of 6, hemoglobin is into the 80% already. So it's, as long as you provide the hemoglobin with a good level of oxygen, it seems to be able to associate with as much of it as possible. So it makes it very efficient as long as you provide an environment where there's a high partial pressure. It's going to be able to do its job really well. So let's explain why that's happening, because you can see initially it didn't go straight up. It kind of followed the line of a normal fluid. It's only around this point here that it starts to get steeper. There's a reason why that's happening. So if we go back to hemoglobin and these heme groups, like I said before, these heme groups are close to the center of the hemoglobin molecule, which means oxygen is having to diffuse further to get in. Now, as soon as oxygen associates with one of these heme groups, so let's say the oxygen is associated with this one, the shape of hemoglobin changes. And we say that there is a conformational change. And when that shape changes, the remaining heme groups become more exposed. And as soon as they become more exposed, it's easier for the following oxygen molecules to associate with. So as long as that initial heme group has associated with the oxygen, the others will be able to very quickly associate with the remaining oxygens as well. And that's what's explaining this sudden increase in steepness. Okay, so let's have a look at this. So the hemoglobin dissociation curve is always S-shaped. Percentage of hemoglobin saturation with oxygen is not directly proportional to partial pressure of oxygen. If it was, you'd expect this straight line. That's not what's happening. It's very difficult to achieve 100% saturation of hemoglobin. So usually you'll see that it gets up to about the 98, 99%. Just that 100% at the end is difficult to achieve. The conformation of the hemoglobin molecule changes as oxygen molecules become associated with it. This changes hemoglobin's ability to associate with further molecules. So like I just said before, as soon as one oxygen molecule binds with one of the heme groups, a conformational change will occur, which will expose the remaining heme groups, and it's going to make it easier for the oxygens to associate with the remaining heme groups. Okay. Last thing to be aware of with hemoglobin, I'll just talk about one more thing as well here. So you can see here, as long as there's medium to high pressures of oxygen, hemoglobin is really good at associating with oxygen. But when you get down to the lower pressures, hemoglobin is not good at associating. We can see that it's not doing a good job at associating. They might think that's a bad thing, but that's actually a good thing. Because what's happening here then, if it's not associating, oxygen is, well, the hemoglobin is dissociating. Oxygen, or letting go. So why is that a good thing? Why is it good that hemoglobin dissociates from oxygen when there's low partial pressures of oxygen? Well, let's have a look at where there's low partial pressures. The working muscles, the respiring tissues, this is where there's low partial pressure of oxygen. When hemoglobin arrives to this region here, it's beneficial that hemoglobin dissociates from oxygen, because you don't want the oxygen to arrive to the biceps and to not unbind from the hemoglobin. It needs to be able to let go of it so that the oxygen can get to the biceps so it can be used for respiration. So it works out both ways. Hemoglobin is very good at associating with oxygen when there's high partial pressures, and it's very good at dissociating when there's low partial pressures. What a perfect situation. It's good at picking up oxygen, and it's good at letting go when it needs to let go, where it's needed. Okay, so last thing to talk about is the types of hemoglobin. So we all have adult hemoglobin. Even a baby within a few days of being born, we say it has adult hemoglobin, because there's only two types. You've got adult hemoglobin and fetal. And fetal is, we're talking about the baby that's forming inside its mother. So if you look at this graph here, fetal hemoglobin is better at associating with oxygen at the same partial pressures compared to adult hemoglobin. So let's look at, it doesn't matter which one we look at, but let's just go with 5, shall we? No, we'll go with 2.5, with partial pressure of 3. At partial pressure of 3, normal adult hemoglobin, i.e. that the mother's hemoglobin, is at around this percentage. What's that, like 8 or 9%? Whereas the fetal hemoglobin is almost double that. So that's really, really important that happens, because fetuses don't breathe for themselves. The only way that they can get oxygen is from their mother. And the way they get that oxygen, I'm going to draw a really bad picture now. I'm going to do it. There's the baby. And the baby has an umbilical cord from this point here, okay? The baby's job to stay alive in its mother is to pump its blood out of its body to the end of its umbilical cord. And the umbilical cord, the other end of it, is connected to the placenta. So the baby's got its own heart pumping. It's not breathing. It's pushing its blood with its heartbeat out to the end of its umbilical cord, and the blood goes to the placenta. And at the same time, the mother's blood, she's pumping her blood to the same placenta. Now the bloods, they're not actually making any contact with each other. There's a very thin barrier being provided by the placenta, but that barrier allows diffusion of small molecules to get by, and one of them is oxygen. So because the placenta is nowhere near the lungs, we would say that the placenta has a low partial pressure of oxygen. And at low partial pressure, the mother's adult hemoglobin is not very good at holding on, or you could say it's very good at dissociating. So the mother's hemoglobin is letting go. It's dissociating from the oxygen. And that oxygen can now diffuse to the other side of the hemoglobin, to the other side of the placenta where the baby's blood is. And because the baby's blood has a higher, we say it has a higher affinity to oxygen, and higher affinity means, let's write that down so we've got that. Higher affinity, the baby's hemoglobin having a higher affinity means it can associate easier, associate with oxygen easier. Which is not well done, sorry about that. Right, as we can see that. So that's really important for the survival of the baby. If the baby's hemoglobin was the same as the adult mum's hemoglobin, there'd be no ability for the baby to pick up the oxygen or associate with it. So this is all completely to do with the survival of the baby. As soon as the baby's born, some babies, their skin will start to go yellow. They're getting what's called jaundice, because what will happen now for the first couple of weeks of their life is they're having to break down their blood cells and break down their fetal hemoglobin. And when you break down hemoglobin, you're breaking down protein, and that process, it needs a lot of excretion. That's the only way you can get rid of all that excess amino acids, which have been converted into urea, is for the baby to be urinating it out. And it takes a while for these bodily functions to be kicking in. So one of the things you can do with the baby when this happens is you can actually leave them out in the sun, and UV helps the baby to undergo this process as well. Right, so there's quite a lot of information there. Let's quickly go back to the first part. So we looked at this molecule called hemoglobin. It's a protein made up of 4 different polypeptides with prosthetic groups called heme groups, and their job is to carry oxygen. Hemoglobin, there's about 280 odd million of these per blood cell, so they've got a really good ability to carry a lot of oxygen. We talked about this idea that hemoglobin has a high affinity to oxygen, and that high affinity is because of the iron, and that fetal hemoglobin has an even higher affinity to oxygen. One thing I didn't mention is this word here, oxyhemoglobin. A hemoglobin molecule which is carrying oxygen is called hemoglobin. So hemoglobin plus oxygen equals oxyhemoglobin. We then talked about the oxygen dissociation curve. And just as an illustration, it shows how at low partial pressures, hemoglobin is very good at dissociating from oxygen, which is beneficial because it needs to let go at respiring tissues. And it's very good at associating when the partial pressures increases. So it's going to be very good at picking up oxygen at the lungs. So one thing just to emphasize, I'm trying to make this simple. I'm using terms like picking up, but I want you to, when you write your essay answers, use the term associate and dissociate. Get into that habit. Don't use picking up, dropping off. It has to be associate, dissociate.